Hi, yeah, my name's Michael Rach. I'm the business unit here at Fluke, business unit manager here at Fluke Biomedical, and I used to be the product manager of the ProSim product line. So thank you all for joining. I'm also joined here by Hongbo Chen, and I'll let him introduce himself as well.
Hi, my name is Hongbo Chen, and I'm a product manager at Fluke Biomedical, and I'm the product manager of Patient Simulator.
So today we're here to talk about pulse oximetry, and I think it's a good place to start if we go through what pulse oximetry is. Essentially, it's the measurement of the oxygenation of arterial blood. Pulse oximetry monitors something called SpO2, and the acronym SpO2 stands for saturation of peripheral oxygen. The amount of saturation means how much of the arterial blood is carrying oxygen. So 100% saturation would mean all of it is oxygenated. The pulse oximeter reads out in percentage of the arterial blood that is oxygenated. So you're looking at ranges, somewhere between 0 and 100%. Biomedical equipment professionals should be well grounded in the fundamentals of this very valuable technology in order to understand it adequately. So in order to do that, we're going to start with the basics, and then we'll build up to cover all of the details. So arteries carry oxygenated blood throughout the body. Arterial blood comes from the lungs, where it has picked up oxygen, and it gets pumped through the left heart chambers to the aorta and then to the arteries. The arterial blood moves in pulses as the heart beats. So pulse oximetry measures the oxygen in these pulses of arterial blood, and then they get to a peripheral part of the body, typically a finger. A pulse oximeter can work quickly, right, getting a result in just a few heartbeats. But it checks that arterial blood is indeed carrying oxygen. So pulse oximetry can be measured with multiple types of devices that can attach to a finger, a forehead, a foot, or even other areas. So many of these are actually becoming disposable over time as well.
Yeah, Mike, just want to add to this, there are some fingertip pulse oximeters, and sometimes people use those pulse oximeters at home. Just recently, a friend of mine got COVID-19 virus infection. Unfortunately, his wife bought a fingertip pulse oximeter to monitor his oxygen level, and one night, and she noticed that his oxygen dropped below 90%. So she took him right away to emergency room. So it's a very quick way to monitor how bad your infection is. So it's getting very popular even at home, not just hospitals.
Yeah, COVID-19 has definitely increased the importance of vital signs measurement technologies like pulse oximetry. First, I'd like to walk you through how the human body metabolizes oxygen. And if you look at this diagram here on the left, it all starts with oxygen being inhaled, right? So the oxygen being inhaled travels to the lung, and then the lung transfers the oxygen into the bloodstream. The bloodstream then has to be pumped through the heart down to the extremities. In the case of pulse oximetry, your finger, your forehead. And then what happens is that oxygen gets used and gets turned into deoxygenated blood. That deoxygenated blood goes back to the lungs. That waste gas, in this case CO2, is exhaled. And then the whole process starts over again. So capnometers and end-tidal CO2 monitors, EtCO2, they measure breath by breath, right? So this is measuring how the body's metabolizing oxygen into CO2. And this is a more instantaneous measurement of oxygen metabolism since it is breath by breath. Any changes to the patient condition will be delayed in a pulse oximeter since enough blood needs to be pumped from the lung to the heart to the finger. So essentially, what you should take away here is that end-tidal CO2 measures how much waste gas is leaving the body, or is the oxygen being metabolized, and SpO2 measures how much oxygen got into the bloodstream. So in the red blood cells, there's a protein called hemoglobin that can carry oxygen to the body. In blood, the hemoglobin can be oxygenated or not. A pulse oximeter measures the percentage of hemoglobin in arterial blood that is oxygenated. So pulse oximetry is a very important measurement for patients, and it gives a quick determination if patients are having trouble getting oxygen into their bloodstream. So if they do have trouble, it can be seen on the pulse oximeter, and it can be treated as soon as possible. Before pulse oximetry, there was no quick way to analyze the oxygen content of blood. They could do it invasively by drawing real blood and then running tests on it, but that took time that patients couldn't necessarily afford, especially when they're on the operating table or under anesthesia. These patients' systems are under stress. So before pulse oximetry, thousands of patients died of undetected hypoxia, which is a low level of oxygen in the blood. And now the pulse oximeter is ubiquitous and can be found in most hospital rooms and emergency facilities. Blood oxygen is routinely measured, and doctors and nurses now know immediately whether the blood oxygen is low. So the pulse oximeter is a good device to have available, and I think now would be a good time to kick off poll question number 1.
Yeah, also I want to add there that it used to be invasive. You have to get your blood, analyze your oxygen saturation. But now with this pulse oximeter, you do that non-invasively. You get the reading right away. So it's a huge improvement.
So I put the poll up. We'll let it stay up there for a minute or so, but do plug in your guess here. There will be a reference in the answer to high school science, so we'll see how far back in your brain you guys can dig. Okay, it looks like people are circling in on one answer, which is good, but there are guesses across the board. All right, so just under 10 seconds left, and I will close the poll and share the responses. All right, so I'll close that, and let me share the answers here. Okay, so it looks like everyone kind of zoomed in on 400 to 1,000 nanometers, which is the correct answer. So good job. Let me hide this. Okay. So the light we see isn't all the light that exists or all the wavelengths that exist. It's rather just a portion. So again, if you go back to high school science class, you might remember Roy G. Biv, which was the acronym for the visible light spectrum. So red, orange, yellow, green, blue, indigo, and violet. Pulse oximeters typically use red and infrared light. So I'll walk through how the most common oximeter configuration works. So the oximeter shines 2 different wavelengths of light through the patient's finger using light emitting diodes or LEDs, one at a time. And those 2 wavelengths are red at around 660 nanometers and infrared from between 900 and 940 nanometers. So congrats to those of you that got it right. Pulse oximeters shine light basically through the human body. Typically it's a finger, but it could also be feet, earlobes, and foreheads. And there are also some oximeters that reflect light off the body, reflective SpO2, and they do that rather than shine light through it. We'll get into that technology a little bit later. But a detector on the other side of the finger will measure how much light passes through the finger from each type of light. The oximeter only needs one detector that can detect either light because the oximeter knows which light is being shown at any particular time. Here's how light is absorbed by the hemoglobin of those 2 wavelengths, right? So oxygenated hemoglobin absorbs more infrared light and allows more red light to pass through. Deoxygenated hemoglobin is the opposite. It allows more infrared light to pass through and absorbs more red light. And you can see that in the graph there. So what's not absorbed shines through to the detector and gets measured by the SpO2 monitor. The SpO2 monitor measures the amounts of red and infrared light detected. It then calculates the ratio of red to infrared light, and that ratio tells it the percentage of saturation. So that's the beginning of the formula here. There is a little bit more to this equation because there's more than just hemoglobin that the light has to pass through when it comes to a finger. There's the rest of the finger, which includes venous blood, tissue, bone, skin, and some other things. SpO2 monitors are interested only in the transmission ratio caused by the hemoglobin and not by everything else. So to do this, they break it into AC and DC components. AC is the varying level caused by the arterial pulse, and DC is the average level caused by everything in total, including the arterial blood. The DC level is the average measurement over the entire time, whereas the AC level is the change from the steady state level between pulses to the level during a pulse. So one is the delta and the other is the average over the entire time. These AC and DC components get combined into the final overall calculation as is shown on this slide. And by calculating the ratio in this way, all the variable factors in the finger are eliminated, such as skin color, finger thickness, and the amount of blood pumped. So SpO2 monitors use a lookup table of ratios called an R-curve. So R-curves have been determined by clinical trials by the manufacturers. And since there's no scientific reference available to characterize the human body, the manufacturers have experimentally measured the ratios that will be obtained at known saturation levels. They put these values into the R-curve lookup table, and that causes this R-curve that you see on the slide. The SpO2 monitor calculates the light ratio and then looks up the saturation level that corresponds to it in the R-curve. And that's what you're going to see on your SpO2 monitor. So with that, I will move. Go ahead, Hongbo.
Yeah, every OEMs does their own clinical trials to come up with different R curves. This is why, you know, in those patient simulators, you can choose different R curves corresponding to different OEMs or brand of oximeters.
So we'll get into that in a little bit more detail in a couple slides, but I want to launch the next question. So I'll give you guys another minute. This one is true and false. Share those results with you. So 69% of you chose false, and that is the correct answer. And I will hide this, and we'll get going on why. So it is an important point to note that these testers are not true simulators, like the ProSim8 itself, right? The FDA has in fact declared that they should not be called simulators. A finger simulator would have to actually have the light shine clear through it and actually contain a pulsing medium that would mimic the actual hemoglobin being oxygenated at various levels. And you can imagine the complexity behind that. So Fluke testers pretend in a different way to be a human finger. And it's done in a way that it can be programmed for a wide range of variables in order for you guys to be able to do the most comprehensive testing available. Fluke Biomedical calls these devices functional testers because that's what the FDA calls them. Fluke does list them in its catalog along with its patient simulators. However, don't be fooled, right? The ProSim Spot is an accessory to a patient simulator, but it is itself a functional tester. So if you want to learn more, there is a Fluke Biomedical white paper called "There's No Such Thing as an SpO2 Simulator" that's on the website. And it's a good read if you're interested in learning more about that. But from a Fluke Biomedical perspective, SpO2 monitors can be tested using a Fluke Biomedical ProSim Spot or Spotlight. And the ProSim Spot is a plug-in module to the ProSim8 patient simulator, and they work together, right? So ProSim8 provides the user interface, and it's the most fully featured tester, whereas Spotlight is a standalone tester with a little bit less capability than the ProSim Spot. These are the latest that we have here at Fluke Biomedical, but we'll get back to the content.
Yeah, so I just want to add something here. It's impossible to make a real human finger because, you know, human finger is so complex. It's not just the hemoglobin. There are all other tissues there. Like, yeah, so this is why, you know, there is no such a thing of a human pulse simulator. It's just a function tester.
No, it's a great point. And as we get through a little bit of the variability that we're able to do with these functional testers, you'll just thinking about how to make that human finger becomes more and more impossible.
Mhm.
So Fluke SpO2 functional testers pretend to be a finger, right? You can see on the left arrow, right, how light would normally flow straight through a human finger. Functional testers, even though they're not simulating a finger, they can fool the SpO2 monitor into thinking that it's monitoring an actual finger. They take the light input from the pulse oximeter, run it against an R curve, and emit the right amount of light to the detector. Um, and you can see that on the right there. The tester has two light detectors with a filter over one of them, so it can discern the type of light, whether it's red or infrared. And as the monitor shines light onto the top of the finger of the tester, red and infrared light in succession, the tester will do a few things. It'll measure the amount of light, it will decide which type of light it is, red or infrared, and it'll calculate for the test settings how much of that light would be shining all the way through an actual finger. It'll turn on its own LED on the bottom so that it looks like it's shining through the finger with that exact amount of light. So this LED has a wide enough bandwidth of light to be detected by the monitor, and there only needs to be one light detector in the monitor because it can detect either wavelength, and the monitor itself should know which wavelength it's outputting at any given time. So the tester is running heartbeat pulses that approximate the shape of an arterial pulse as well. It varies the light according to the current amplitude of the pulse. So in between pulses, it gives just the DC light, and during the pulse, it gives the AC pulse plus the AC light plus the DC light. So it's a little bit complex, but essentially that diagram on the right is what a functional tester is doing. So why not test with your finger, right? Your finger can test the oximeter only for the state of your finger, which is unknown to you, right? A typical healthy patient has a very stable SpO2 reading in the mid to high 90s in percent. And a tester actually allows you to vary the settings across the physiological range of variability, right? So oxygen saturation, right? Through the range of actual percentages that could be encountered, right? So think about you having to simulate different percentages with your own body. You would have to actually starve yourself of oxygen, right? Heart rate, you'd have to change your own heart rate to go from slow to fast. And there are numerous others that we'll touch on in other slides, but your finger just can't do that, right? So in Prosumate with Spot, all the heartbeat signals are synchronized, right? So the ECG and the blood pressure beats are all on the same beat and timed to be physiologically accurate. In fact, the SpO2 signal is derived from the arterial blood pressure beat, delayed, right, to represent the actual delay of the pulse traveling down the arm to the finger, right? So having that be synchronized helps you have an even better simulation, right? So people always confuse transmission and perfusion, and I'm sure this isn't any different for many of you guys on the webinar today. Hey, Hongbo, do you know the difference?
Yeah, like you said, people always confuse with the difference between transmission and perfusion level. And transmission is basically is a percentage of light, as you show in that on the slide, goes through the finger. Different people have a different fingers, thickness of the finger, different colors, yeah, a neonatal food. So that transmission basically reflect that differences.
Absolutely. Yeah, the SpO2 monitor has to be able to account for all different types of fingers, right? And to make sure that the oximeter is working for all types of patients because we're all different. Um, Patients are being diagnosed and monitored using these pulse oximeters. So it's critical that these readings are correct, not just for a typical finger, but for all fingers, right? So Again, if you look here, the settings there are for a dark thick finger, a medium finger, a light thin finger, a neonatal foot. They're all different, and we have to make sure that it works for across the entire range.
Yeah, that's another reason why you should not use your finger as a simulator to test your oximeters, because you're just one type of finger versus many other different fingers. And yeah, you should test it with a simulator with all different settings.
That's a great point, and I know a lot of people will slip on a glove, a latex glove that's purple to simulate darker skin tones. But again, you're limited in what you're actually testing. One, you don't know what you're actually testing, what variable you change. You just know that it should be different. So here you get to control that variability, and you do get to change, test a wider range, and it's repeatable, right?
Hey, Mike, just a very interesting question for you. So, people use fingers to put on the sensor, right? Do you know normally which finger they're gonna put into the sensor?
I would
Just
think
a
the
guess.
pointer finger, right?
It's actually yeah, index. 80% people use their index finger to do the test.
I'm looking at my hand now and wondering if I would ever use a different finger. So that's a good point.
Yeah.
So there's another term here called pulse amplitude, which is also known as perfusion. And that's the amount of arterial blood delivered to the finger in each heartbeat. So people confuse these two terms as well, but they mean the same thing. So during a heart pulse, there's less light that will be transmitted because of that additional blood in the finger. So this setting in percent is how much the light level varies from the average DC level, right? So this setting tests how well the monitor works in the difficult situation where the patient is not pumping very much blood, right? So you're able to change the amount of blood in the finger here, which again, you wouldn't be able to do if you were using your finger. Another thing you can vary with a functional tester is the ambient light artifact. This is a setting of extra light that could be in the patient's environment or the patient room, right? That could interfere with the SpO2 monitoring. These settings, are outdoor sunlight or indoor light of various frequencies that might be encountered. Fluorescent lighting is the closest in wavelength to the red and infrared light being measured by the pulse oximeter. But there's incandescent, there's screens, there's LEDs, there's lots of light in any given space. The amplitude of the extra light can actually be set up to 5 times the amount of transmitted light. In the monitor, this is again, you're trying to make sure that the monitor should be able to work even with the extra light in the area. So again, just another variable that you're testing across. There's another artifact called Respiration Artifact. And this is a variation where the patient's breathing can affect the SpO2 pulse, right? Similarly to how it can affect blood pressure, this breathing, right, can change the SpO2 pulse amplitude during each breath. So during maximum inhalation, there's a pressure on the chest that can reduce the amount of blood pumped for each heartbeat. So again, the SpO2 monitor should reject the effect of respiration and give stable readings. This is something you can also simulate with a functional tester. So since all SpO2 monitor manufacturers have developed their own R curves, they vary a bit, right? They're not the same. So the components used in the monitors, such as the light emitters and detectors, can differ from each brand and model. And on the tester, the user must select the manufacturer in order to use the correct R curve. So Fluke has worked closely with some manufacturers that have supplied their R curves to Fluke. And for other manufacturers, we've figured out the R curves experimentally, right? So we've tested to see what ratios it took to get specific SpO2 percentages on the monitor, and we've programmed those in to our functional testers. So manufacturers are now using light to monitor not just SpO2, but other parameters. Notably, Massimo Rainbow, their oximeter uses 8 wavelengths of light for the additional monitoring of hemoglobin, right? There are some hemoglobins that cannot carry oxygen, right? And there's an important situation when carbon monoxide gets bound to the hemoglobin, right? Called carboxyhemoglobin, right? And another is where the iron molecules are not the right type that carry oxygen, right? So this is methemoglobin, right? So Massimo Rainbow can detect the percentages of these hemoglobins as well. So again, trying to do more with light. Massimo has worked with Fluke to figure out how to test the rainbow. And Massimo has designed a special cable that interfaces to the finger sensor that clips onto the Fluke biomedical tester. They've approved this test to be adequate for Rainbow to be tested by Fluke biomedical testers. And these detectors on our side can just detect the 2 wavelengths of light. But again, Massimo has approved this for use. So do use something that the OEM has backed. So there's a couple trends in pulse oximetry. One is reflective SpO2. So reflective light technologies don't shine through the finger. They shine light onto the tissue and then measure the light that's reflected back onto a sensor. So on that image on the right, instead of the light passing straight through the finger on the left side, it actually gets shown onto the finger and it gets reflected back. This technology is growing and it's becoming more and more ubiquitous in the market. It's easier to build pulse oximeters into personal devices, right? If you do a quick search of the internet, you'll see all the many options that are out there. And this is a trend that we should expect to continue into the future. We should expect more light monitoring technologies to emerge, and we should expect light to be able to measure even more body parameters going forward. Hongbo, I know that you've been on some interesting calls with some startup companies. You want to share any of those examples?
Yeah, they're like, make all kinds of new pulse oximeters, used on smartphones and yeah, on their wrist while they're doing exercises. So there are a lot of advanced technology in this field. Yeah, we are trying to make in the future, make a patient simulator do all kind of testing. Right now, the Person 8 does not do a reflective light technology. Hopefully, in the future, our next generation of pulse oximeter can test reflective light technologies.
Yeah, I think this will be a really interesting space to keep an eye on and see where it grows.
Mm-hmm.
So we do have one last poll question that I do want to throw out there. So I'll launch this one. This is just a quick yes or no question. I did like the battle in the last true and false. So let's see where this one heads up. Okay, so we're 50-50 with 5%. We'll keep this one running. Oh, there's an interesting little back and forth going here. Oh, it's close. All right, so we got about 20 seconds left. Back at 50-50. An undecided group out here. Oh, it's only a 6-point spread. 10 seconds to go. All right, so we'll wrap it up in the next 5 seconds. All right, so let me close this and share it. So 56% of you answered yes. And that is actually the wrong answer. So those that answered no, you guys got that one right. So let me get to the next slide. So 44% of you were right? You cannot calibrate an SpO2 functional tester. this is due to the fact that there's no primary calibration reference to calibrate it against, right? So if you think about how this is built, you are taking light, you are then applying it to an R curve, and then emitting light. So there isn't anything that you can really measure that against. What Fluke Biomedical does recommend is that you send it in for an annual validation, right? That makes sure that everything's working properly, but this isn't a calibration. Another thing I would note is that if you want increased accuracy, we mentioned those manufacturer approved R curves. It's recommended that you do use those, as these have been approved by the medical device OEM themselves. So that's a way that you can have even more confidence in your test results. But again, do send them in for a validation, and we'll make sure that the device is working properly. But again, there's no calibration because there's no calibration reference for a functional tester. So in summary, pulse oximetry is a very important technology that saves lives, right? And it's destined to become even more common with the use of personal monitors and reflective SpO2 and wearable devices. So given today's situation with COVID-19, in the current use case of pulse oximeters, this could be and is definitely a key leading indicator of infection for patients, right? And it's all the more reason that we should be ensuring that our pulse oximeters are operating the way that they should, because this is how our COVID patients are being diagnosed and monitored. So I do urge you to make sure that your pulse oximeters are working correctly. And yeah, it's definitely more important given today's situation. So there's a couple ways that you can learn more. One, you can reach out to your local sales rep and get a demo. You can actually play with some of our devices and learn what they look and feel like. Other ways is you can visit our website, right? This is where you can find the app note that we touched on earlier. And we're also really active on YouTube. So YouTube has quite a few videos that you can watch to see some of our functional testers in action and how they can work with many of our other devices. LinkedIn and Facebook is a good place to tune in and find the latest from Fluke Biomedical. And of course, Advantage Training, if you want on-demand training for many of our Fluke Biomedical products. So with that, I just want to thank everyone for joining today's webinar. I appreciate you guys taking the time to learn a little bit more about SpO2. And again, do make sure that you test, especially during these COVID times. It's becoming increasingly more important. So let's do all that we can to ensure patient safety.